And I even learned to unmute. It only took 2 years of COVID. So thank you for the invitation to speak here. And today I'll be talking about using C. elegans to understand host-pathogen interactions. So why do we care about host-pathogen interactions? Well, as our medicine and healthcare develops, unfortunately, bacterial infections becoming more and more threatening. The number of hospital-acquired infections will growing steadily over recent decades, and now it's more than 1.7 million nosocomial infections that lead to about 100,000 deaths. And this is actually the main source of premature death worldwide. The problem with bacterial infection is that we use antimicrobials to kill bacteria, but at the same time, bacteria develop mechanisms to be resistant to these antimicrobials. Sometimes it takes several years to develop such resistance, but sometimes it's only a matter of a few months. However, the overall story is the same. For every antimicrobial that's been introduced into clinics, and broadly speaking, for every class of antimicrobials, there is known resistant mechanisms that is developed. And unfortunately, for these and other reasons, we see decline in the amount of new antimicrobials on the market. Obvious targets already exhausted. It's hard to come up with novel chemical scaffolds. It's also very expensive to develop a new drug, and then either bacteria will develop resistance if this drug is used widely, or this drug has to be used as a therapy of last resort, and then it's not profitable for the company. So these and other reasons led to the decrease in the production of new antimicrobials over the recent years, as you can see, while the rise of antimicrobial resistant bacteria keeps occurring. And just quick example here, since you're somewhat microbiology proficient audience, just one small piece of information. In 2015, there were about 6,700 infections with multi-drug resistant Pseudomonas aeruginosa a year, and less than 500 deaths. However, just 4 years from that, we see these numbers increasing 4- or 5-fold, from 6,700 to 32,500, with corresponded 2,700 deaths. Of course, this is a huge toll, not only in patients' lives and health, but also for financial burden for the country overall. Therefore, since it looks like we are not going to develop a new golden bullet antimicrobial that can be efficient and not cause resistant development, we should consider alternative approaches. And one of these approaches is to inhibit bacterial virulence. Different bacteria express a quorum sensing system where they can produce molecules to communicate with each other. They make biofilm that prevents their killing by antimicrobials. They have various secretion systems, some of which allow to inject protein and other toxins directly into the host cell, damaging it and eventually killing it. Additional toxins may be released into the media. One of the ideas behind antivirulence is, let's start with this dangerous pathogen with wide armamentarium of toxins and other virulence factors, and then we'll just block their production. This way, bacteria is still inside of the host, but we're forcing it to acquire a commensal-like state under which not much of a host damage can occur. And this is particularly important in case of drug-resistant and multi-drug resistant bacteria, because these infections may take a long time to be treated, as different combinations of antimicrobials have to be tried. So if in the meanwhile we block the damage that pathogen does to the host, we give patients better chances to maintain health until proper antimicrobial combination will be designed. However, to inhibit antivirulence, we need to be well aware of important virulence factors for each infection. And for this, we need to study not bacteria by themselves or immune system of the patient by itself, but together a host-pathogen interactions. C. elegans, Caenorhabditis elegans, a small roundworm, is a great model organism to study host-pathogen interactions. Worms have powerful genetics, both forward and reverse. Also, their size allows them to easily fit into free atypoval plates, enabling high throughput applications. Worms are bacteriivores. Normally, we feed them E. coli, but if we want to develop a pathogenesis assay, then we just need to find a suitable media and substitute E. coli with a pathogen of interest. Worms are transparent, which makes it easy to follow activation of immune pathways if we'll target downstream effectors with fluorescent proteins. Worms do not have specialized immune cells. They have what's similar to epithelial immunity, and I think you'll hear more about it during the second talk today. Another important fact to consider is majority of human pathogens can infect and kill C. elegans, and that include gram-positive, gram-negative bacteria, fungi. Well, viruses are more specialized. There are several viruses that kill C. elegans, but they worm-specific. Importantly, these pathogens frequently use the same mechanisms to kill the worm as they use in infection of a patient. So by studying C. elegans bacteria interaction, we can identify relevant virulence pathways and relevant defense pathways that could be translated to human infections. And the black line here just shows C. elegans infected with gram-negative bacteria Pseudomonas aeruginosa, which is the main focus of my lab. And upon infection, worms die in a matter of 3-4 days. However, if they feed their normal food source E. coli, then they live for over 2 weeks. So we can easily differentiate between worms on a healthy food and worms on a pathogenic food. The assays that are shown here were performed on agar, where individual worms are tracked over time. However, these assays are somewhat cumbersome. They require a lot of scoring time, a lot of attention, and have more variability. That's why one of the things we like to do is to move assays to liquid format when they're trapped inside of free atypoval plate and cannot leave it. And also we can standardize conditions more easily. We have machine like fax, but just for worms, called worm sorter, that we use to dispense exact number of worms into each well of free atypoval plate. Then we can add the bacteria of interest, Pseudomonas aeruginosa, wild type, various mutants, or Pseudomonas plus drugs, and let infection proceed. After a certain number of days, usually about 44-48 hours, we'll stop infection by washing plates to remove bacteria. At the same time, we're adding fluorescent dye, and after that, image plates on automated microscope. Then we can take images in bright field and in fluorescence. This dye has properties that it can only enter nucleus and stain DNA if membranes are damaged. So when worms are alive, their membranes are intact. And that means virtually no staining is observed. If worms die, then membrane damage leads to a robust staining. By analyzing fraction of fluorescence, we can infer the fraction of death. And here is an example when worms were grown on E. coli, they alive, happy, curvy, and when they were exposed to Pseudomonas aeruginosa, they dead, 6. After we developed this liquid assay, we wanted to know which virulence mechanisms play a role in the host killing. So we analyzed the number of usual suspects. Here you see worms treated with E. coli, and each well has identical number of worms, but you can only see dead fluorescent ones. Here they're alive. Wild type Pseudomonas, pretty much all of them are dead. Mutants in quorum sensing, pellicle production, phenazine production, so that would be your biofilm, or cyanide production, did not show any attenuation. However, when we use mutants in pyrogen biosynthesis pathways, or PVD mutants, we see decrease in host death. And these are bars showing quantification. So in the presence of pyrogen biosynthesis mutant, the worm death rate dropped twofold, about twofold or so. This is how we got interested in pyrogen. Pyrogen is a siderophore. It's an iron scavenging molecule that's made by Pseudomonas and secreted into the media. It's synthesized non-ribosomally and has this really interesting chemical structure. Pyrogen is fluorescent. We can very easily track its presence in the media or on a plate. It was known that pyrogen can activate multiple virulence factors, and it's also required for biofilm formation and necessary for Pseudomonas full virulence in mice. However, all these roles were considered to be indirect due to the pyrogen role in iron metabolism. From our collaborator, we obtained a number of isolates from a sputum of pediatric patients with cystic fibrosis and test for their ability to produce pyrogen. We noticed that pyrogen production level vary from very high to nearly non-existent, with our lab strain being somewhat in the middle. But next, we also looked at the ability of this strain to kill C. elegans. And we observed a strong correlation. The higher amount of pyrogen produced, and here they normalized to lab strain, the lower C. elegans survival. And this observation is important because pyrogen production is observed in about 80% of patients with cystic fibrosis. So yes, pyrogen plays a role in pathogenesis, but that was known. The question that we asked was, can it be directly toxic to the host? To answer that question, we prepared filtrates, bacteria-free spent media, where we let Pseudomonas grow in the media overnight. Then we remove bacteria by centrifugation and subsequent filtration and exposed pathogen to that media. What we noticed is that if we prepare E. coli filtrate from E. coli, then worms are alive, just same as media control. If we prepare a filtrate from Pseudomonas aeruginosa, worms die within a couple of days. Iron is pyrogen's natural ligand. If we provide iron to quench pyrogen, that worms remain alive. So this toxicity is related to pyrogen's ability to bind iron, well, possibly within the host. And here, just to show you, we did experiments with commercially available purified pyrogen, and the same thing, we were able to observe host death in the presence of pyrogen that was rescued by the addition of exogenous iron. Pyrogen is an iron chelator. It can deplete iron in the media. However, we thought that if just by altering media composition, it's not likely to kill worms in this time frame. So we hypothesized that pyrogen has to be getting inside of the host and interfering with host iron rather than iron in the media. Luckily for us, it is fluorescent, and it's fluorescent in a near DAPI channel. So we were able to stain lumen of C. elegans with a red membrane marker, so that C. elegans mouth and then starts intestinal tract with its pharynx. And pyrogen, you can see here, fluorescent in blue. This fluorescence is absent from pyrogen biosynthesis mutant or from iron quenched pyrogen, confirming that indeed right here we see pyrogen inside host tissues. When these worms were analyzed and level of iron measured, we noticed significant decrease in host iron in the presence of pyrogen. We were curious, what does it do to the worm? What's the host part of this host pathogen interaction? To answer that question, we performed microarray studies where we exposed C. elegans to whole bacteria Pseudomonas aeruginosa, purified pyrogen, or chemical iron chelator phenanthroline that has completely different structure. We noticed that a significant number of genes were upregulated under all three of these conditions, confirming that likely effects of pyrogen are due to the iron chelation and not something else. And at least the proportion of C. elegans response to Pseudomonas aeruginosa is response to pyrogen. To look deeper into pyrogen specific response, we analyzed the data available from multiple labs where worms were incubated with different pathogens, Staph aureus, Enterococcus faecalis, Candida albicans, plus our data on Pseudomonas. So we had 11 different microarray conditions, and we analyzed the expression of these 200 plus pyrogen upregulated genes. We looked for only one very specific condition with the upregulation specifically at microarray 1, which was high pyrogen Pseudomonas producing conditions. So these about 50 genes were only upregulated in the presence of pyrogen, but not any other pathogen. When we analyze promoters of those genes, as we hypothesized their co-regulation, we found this element being highly enriched. Literature search revealed that this is an ethanol and stress response element, and this element is upregulated by multiple kinds of mitochondrial damage. So here you see C. elegans treated with DMSO, and S3 reporter in these worms is not glowing. In the other slides, S3 reporter is upregulated in the presence of different mitochondria affecting substances like complex 3 inhibitor antimycin A, complex 1 inhibitor rotenone, uncoupler CCCP. So from that, we guessed, since S3 is upregulated under pyrogen treatment, that likely pyrogen does something to mitochondria. And specifically, S3 is known to be activated by the level of a superoxide radical, so by the level of ROS. Other mitochondrial damage response pathways like mitochondrial unfolded protein response or mitochondrial MAPK signaling do not respond to ROS as clearly. So we hypothesized that pyrogen causes mitochondrial damage and that leads to high ROS. One of the ways to do it is to interfere with electron transport chain, because then more electrons can aberrantly go to oxygen, resulting in the production of superoxide radical. So here we use reporter for complex 1 that will monitor NADH to NAD plus ratio. Normally, complex 1 oxidizes NADH to NAD plus, and there should be virtually no green fluorescence. However, in the presence of pyrogen, we see buildup in NADH, which correlates with complex 1 dysfunction. Dysfunctional mitochondria are targeted for degradation by autophagy of mitochondria, specifically called mitophagy. So right here, when they get damaged and have sustained depolarization, they targeted for this degradation. We used reporter for mitophagy regulator kinase pink1, and red is just a transgene marker. So our target gene is in green, and we see much less mitophagy happening normally, which is reasonable, and its level strongly increasing in the presence of pyrogen. We can further confirm mitophagy by using mitochondria-targeted RFP-GFP fusion, where GFP is pH sensitive. It fluoresces in cytosol, but not in autophagal lysosome, while RFP is always fluorescent. So normally we see both red and green signal, but if worms were treated with pyrogen, green signal decreases, which is correlative with mitochondria being targeted to autophagal lysosome for mitophagy. We also observed decrease of mitochondrial DNA and decrease of ATP. So in this simple system, we were able to identify pyrogen as a causative virulence factor that's secreted by Pseudomonas it gets inside of C. elegans and removes host iron. That leads to mitochondrial damage and increase in ROS. Also an ADH buildup that causes reductive stress. And this mitochondrial damage leads to the upregulation of S3 mitochondrial surveillance pathway. And just at the end, briefly 1 minute of how it's relevant to mammals. Turns out Pyrogen doesn't just kill C. elegans, it can also kill mammalian cells. Here you see macrophages that have much lower survival in Pyrogen-containing filtrate than in analogous one prepared from Pyrogen-deficient mutant. Similarly, we see Pyrogen in blue inside of macrophages, and upon Pyrogen treatment, we observe mitochondria fragmentation similarly to what was seen in C. elegans. And here my last slide, moving from worms to cell culture to now mice. After we use C. elegans to identify high and low virulent cystic fibrosis isolates based on their Pyrogen production, our collaborator tested them in mice and noticed that those that didn't make Pyrogen couldn't kill mice, but Pyrogen producing strains were highly virulent, and that virulence were correlative of the amount of Pyrogen produced. So we are working on other aspects of this project. For example, we found drugs that inhibit Pyrogen and they rescue C. elegans and mice, but this is already, time is getting pushing. So if somebody asks, I can tell more about it later. And with that, I want to thank my lab. The research I talked about was mostly performed by Alex Tang, first undergraduate and graduate student in the lab, Elisa Chahiona, who is now a postdoc, and Alexei Riftovich. And that's our funding sources. Thank you for your attention. And now I'm ready for questions.
Great, thank you, Natasha.
This is Heidi. I have a quick question. Hi, Natasha, that was fantastic. I was just wondering, so one thing I didn't quite get with the with your talk, which was great, was is this when the Pseudomonas is secreting the Pyrogen, and it gets into the and it causes there to be an iron sequestration in the C. elegans. Is that limited to some tissue? Like, is it just in the intestines, or does it, you know, do you see it progressing into other areas?
It's not just the intestine. Unfortunately, ICP-MS gives us overall result, but I know at least by microscopy, we can also look into mitochondria in muscles where they're really nicely oriented. And in the presence of Pyrogen, mitochondrial network breaks down. So it's not just intestine, it's also muscles. Neuronal cells are small, we just can't observe mitochondria there easily.
But in the native state, do you just see it where the Pseudomonas is, or, you know, I can see that if you add Pyrogen to the C. elegans, you might see it, but in the native state, when you're just using the Pseudomonas to provide the Pyrogen, is that limited then or not?
So when we're just using Pseudomonas, that would be harder because Pseudomonas phenazines autofluoresce too. It will preclude us, then the whole worm says like bluish greenish from phenazines and its own intestinal autofluorescence. We just cannot do microscopy, unfortunately. So we have to use purified Pyrogen. It's cleaner sample.
Yeah, okay, great. Okay, thanks, it was fantastic.
I have a question. So if you block autophagy from happening, does it suppress or enhance the death of the worms?
Sorry, what suppresses or enhances death worms?
If you block autophagy.
If I block autophagy, it enhances death of worms.
Enhances.
The response is protective. We tried mitophagy mutants and generic autophagy mutants like LC3 or LG. elegans ortholog, and bacillium and worms die faster. Being able to get rid of damaged mitochondria is helpful, I think, because damaged mitochondria generates too much loss and contributes to death.
Thanks. Great work.